So we left off talking about how humans have 23 pairs of chromosomes. Now, there's an important sort of thing that we have to keep in mind. I'm talking about pairs of chromosomes. So if I wanted to talk about individual chromosomes, then I would say that humans have 46 chromosomes. Now, there's an interesting shorthand that's used in genetics to denote the number of chromosomes an organism has, because this 23 pairs 46 is individual to humans. Each organism is going to have its own number of chromosomes. However, in sexually reproducing organisms, they're always going to come in pairs, because regardless of whether you're a human or a butterfly, you are going to get 50% from mom and 50% from dad. And as we know from being math scholars, you cannot divide an odd number evenly. So we notice chromosomes always come in even numbers. The shorthand that's used to represent this number is N. N is going to equal the number of copies of each chromosome. So humans have 23 different chromosomes. If we want to represent the total number of individual chromosomes, that's going to be represented by 2N, meaning 2 times the number of different chromosomes. So for humans up here, our 2N number would be what? So hopefully you got it. So N is your number of chromosomes. Human have 23 different chromosomes. 2 times 23 gives us 46 total chromosomes. So when we take a look at our somatic cells, our body cells, there is a special name for these cells that have 2 copies of every single chromosome, and that term is diploid. Now, diploid is a word that you are going to hear a lot of in this unit. The prefix di, di means 2. So if you can remember that early on, it will help you much later when we start differentiating between diploid cells and haploid cells. Diploid cells are your body cells. They have 2 copies of every piece of genetic info. So if we look over here, so this image on the side, this is called a karyotype. This is used by genetic counselors to identify any genetic disorders. So if you are missing a part of a chromosome, you've got some extra pieces of chromosome, they can create one of these as an image and take a look at what's going on with your DNA. So we notice that they're ordered from 1 to 23, but we notice that the numbering stops at 22 due to the fact that our 23rd pair, those are our sex chromosomes. And we'll do this again later when we do genetics. But we notice that for chromosome number 1, you have 2 copies. Chromosome number 2, 2 copies. Chromosome number 3, 2 copies. That is what a diploid cell looks like. It has 2 copies of every single different chromosome. So some interesting vocab terms that I want to talk about now that we'll see a lot this unit. So when we look at those 2 copies of the same chromosome, so if I go back here for a second, I'm going to pick on chromosome number 1. So let's imagine for a second that my chromosome over here on the right, which just looks like a straight line, came from mom, and then my chromosome over here, oops, so this is even slightly more to the right, this would be my chromosome that came from dad. So I get 1 whole copy from mom, 1 whole copy from dad, and it's the same info. So if there was info on eye color on this chromosome number 1, I would get some info about eye color from mom and some info from eye color from dad. We call these chromosomes, the maternal and the paternal chromosome, of the same. So my mom copy of chromosome number 1, my dad copy of chromosome number 1, homologous pairs. Homo meaning the same and logos meaning locations. They have the same genes in the exact same spot, but they're not genetically identical. So for example, my dad has brown eyes and my mom has blue eyes. So dad could have given me a copy of brown-eyed info and mom could have given me a copy of blue-eyed info. So that means it's the same information, the same genes on these 2 copies of chromosomes, but it's not necessarily the exact same genetic sequence. We find that when our chromosomes replicate, so when they go through a process known as DNA replication, my dad chromosome and my mom chromosome of chromosome number 1 are going to make an exact copy of themselves. And then they're going to attach at the center. And that's what gives us that nice X shape. So when you think chromosome, you tend to think X-shaped. But what you're actually picturing is you're picturing a replicated chromosome. So a chromosome that has undergone this DNA replication to make an exact copy of itself. These 2 halves of a replicated chromosome are genetically identical. So my 2 copies of dad's brown-eyed information would be on each half of my replicated chromosome. My 2 copies of mom's blue-eyed information would be on my 2 halves of mom's replicated chromosome. Now, these are ideas and terms that are going to follow us throughout the rest of the year. So review them and kind of make sure that you kind of are familiar with this idea of homologous chromosomes being my mom version and my dad version, but not necessarily the same info. and my sister chromatids being my replicated halves of a chromosome that are genetically identical. It's 2 copies of dad's info on one or 2 copies of mom's info on one. So let's see if you got it. So homologous or sister chromatid. So if we take a look right here, I have chromosome A, chromosome B. I tell you that on chromosome A, there is information for blue eyes, and there's information for blonde hair. And on chromosome B, there's information for brown eyes and information for brown hair. So hopefully we were able to get that since we're looking at chromosomes that have the same genes on them, but they're not genetically identical. Those are sister chromatids. So this would be my mom copy of chromosome 1 maybe, and dad copy of chromosome 1. Next one, I have that X shape, and I have 2 copies of blue-eyed info, 2 copies of blonde hair info. Oops. Good, so this would be a homologous chromosome. Sorry, this would be a sister chromatid. Oh my goodness. So this would be a sister chromatid. It's a replicated chromosome, each of those halves. All right, so the last one, we're going to do a little drawing. So in your number 10, you'll notice that it says interphase, and it asks you what chromatin is. So what you're going to do is in your notes, you're going to write down our definition. So it says chromatin is DNA loosely wrapped around proteins, and it's not visible. And then what I want you to do is, you'll notice I have a very lovely drawing that I made on here on the left-hand side. So you're going to sketch what this would look like in a diagram. So what it is the nucleus is drawn as a circle. So that's your nuclear membrane, the membrane that goes around your nucleus. And then inside your DNA is just long, thin strands. If we looked at a cell underneath a microscope and when the nucleus looks empty, it's not empty. It's just that the DNA is so loosely wrapped around the proteins, we can't see it. The next thing we're going to do is underneath, so I'm still in number 10, but I'm in my second bullet. So it says during division chromosomes. When you get ready to divide a cell, or when a cell gets ready to divide, it doesn't leave its DNA all loose and laying around all over the place. It's like if you were going to move houses. You wouldn't move each individual object in your house one at a time. You would put things in boxes, you'd get them all nice and organized, and then you'd move the boxes. DNA does the same thing. So your DNA is going to tightly coil around these proteins to give us that X-shaped chromosome you're familiar with. Now all of the DNA is packed up into these nice little containers, and it's much easier to move it around the cell and divide it between cells. So what you're going to do is jot down our definition. So DNA tightly wound around proteins, and this density of winding them really tightly makes them visible, so we can see them under a microscope. And then you're going to copy down my nice sketch over here. Remember, you can pause at any time. So I have my nuclear membrane, which is my line, and then I've just drawn my X shapes inside as my chromosomes. The last thing we're going to do is we're going to give ourselves a little bit of an idea of the structure of our replicated chromosomes. So this is a chromosome that's made a copy of itself, and it's going to be attached at what we see is called the centromere. So you'll notice in your notes that you've got these labeled down, or copy down, please, these labels in your notes. So the center part right there where your 2 sister chromatids, so each half of this replicated chromosome is called a chromatid. Collectively, they're frequently referred to as sister chromatids. And remember, these halves are identical to each other. They're going to be attached at a point in the middle called the centromere. We call the ends of each of the chromosomes a telomere. Now, telomeres are significant or interesting to us because a lot of evidence shows that the reason that cells know when they need to die or when they're too old and they've been living too long has to do with their telomeres. So the telomere actually breaks down at the end of the DNA. And when it gets to a point where it's broken down too much, the cell is going to know, okay, our DNA is poorly degraded, so we need to, basically, the cell needs to destroy itself. And the last term we need is that coming off of the centromere, each of our segments of DNA that comes up, those are called the arms of the chromosome. So when we look at sort of identifying where genes might be on a chromosome, frequently it'll tell you what arm of the chromosome it is going to be on. So what you should do now, I want you to go through, you should have gone through 1 through 11, should have everything filled in. You can rewind this video as much as you need to make sure you've got it all. On the back of page 1, you'll notice that you have instructions for filling out the cell cycle. So this is how to understand sort of the life of a cell and what are the different parts and why are they important. So please work on that next.